basically here we have, or it is very important to study about viruses, or we can say that. And these viruses are considered as some examples of evolution, basically. So we go in for study of virus, basically it gives us some clue about evolution. And as you told, it exchanges genetic material with the host cell. You talk about the characteristics or reasons why you mean to study is it is a very big evidence of evolution. It exchanges genetic like material with the host cell. Even I think about 7 to 8% of your DNA will be viral. Like that much of exchange happens. So viruses has the ability to evolve very rapidly and adapt to any environment. And so that is the reason scientists usually take viruses, because they give useful insights about the mechanism of evolution. Apart from this, viruses are used for treating diseases like cancer, gene therapy, and even development of vaccine. It is actually dead
Yeah, they
attenuated.
use like
Yeah.
don't they use like the dormant thing and
Yeah, correct. So it's a dead, we call it as dead attenuated. And that is what. So that is what is used to make a vaccine. The same applies for the COVID which we faced recently, right? So it was a COVID vaccine, means from the virus only. So studying these vaccines or studying about viruses is benefiting us in so many ways. And the term virions, have you heard about this term virions?
no.
Okay. So it is a unit of the virus that is present outside the host cell. So just one unit. So it's a basic, like how we call cell as a basic unit of life. Similarly, for viruses, these are the basic unit that is present, and these are particles that is responsible. And maybe we can say for either spreading or to just get into the host cell. You call that particles, the basic unit of the viruses, as virions. It's fine. So these are
Okay.
just some informations that we need to know. And basically, we know each organism, whatever organism it may be, the size and the functions or the mechanism of action, and varies from one to another. It's not the same like for everything. Talking about the structure. So what have you learned about the structure of virus?
We have like a capsid head.
Very good. Capsid is made up of?
Proteins, if I'm not mistaken.
Correct. So proteins. So capsids are made up of proteins, right?
Yeah, then they have the
Okay, so when we
don't have
talk about this,
cytoplasm. They
yes, it has
have,
a cytoplasm. Does it have a genetic material?
yeah, it depends, DNA or RNA.
Yes. So this viruses, as we know, it's microscopic, but usually you can consider the size of a virus to be 20 nanometer to 500 nanometer. So as I told you, these viruses, as you told me, 1 second. Yeah, tell me, did you say something?
No.
Okay. So the viruses contain either DNA or RNA as a genetic material, and you also mentioned about the capsid. What is a capsid? You told me now.
It's a head made of proteins.
The protein shell, which protects this genetic material. Okay. And it is made up of a protein. And it has, some viruses have enzymes, some viruses doesn't have enzyme. So you told me one thing which is not present in the virus. What is it?
Cytoplasm.
Yes. So they do not have a cytoplasm. Or one more thing.
Organelles.
Plasma membrane.
Oh.
They don't have a plasma membrane also. But what about the size of bacterium? Have you learnt about it?
I don't recall.
It's mostly 1 to 10 micrometer, which is approximately nanometers. If you see, I think it is 1,000 to 10,000 nanometers. And you basically know the structure of any eukaryotic cell. So it starts from 10,000 to 100,000 nanometers. So among all the organisms, this viruses, we consider them to be a very small, to be seen with even a light microscope.
Mm-hmm.
Okay, so you use only the electron microscope. Mostly, we use the electron microscope for the study of the structure of virus.
Mm-hmm.
Right? And so we're talking about the structure in little more in detail. It has a diversified structure in the shape and the structure. Not all viruses have the same shape and structure.
Yeah.
So it has a capsid called the protein sheath and it protects the material. Sometimes the capsid which we are talking about may be simple and spherical.
HIV.
Mostly HIV, how does it look like?
It's like the circle with a little like and
No,
like they
capsid
look a bit
like this,
like
right?
enzyme receptors sticking out on the outside
Like
of
this,
it.
something like this. Right. Yes. So influenza virus, some as we know, it is a little complex and has multi-layered capsules also. Oh, and HIV contains what genetic material? DNA or RNA?
I don't know, man. I think this is something I'm supposed to know, but I don't know right now.
Single-stranded RNA. HIV, influenza, even coronavirus all has single-stranded RNA. Have
Ah,
you heard
okay.
about rotavirus?
No,
Hmm?
no.
That was also famous some time ago. Diarrheal disease. It's a diarrheal disease, mostly common in infants. Okay, so this rotavirus is caused by double-stranded RNA. Yeah, I don't think you will have heard about this parvovirus. Say yes or no.
I still don't know. I only know
Hmm. COVID.
HIV, I know corona, I know
Okay, this
influenza.
parvovirus
That's
is an
it.
animal infectious disease, mostly in dogs and all. It spreads from dogs like rashes. As from dogs, it passes on to the humans. It is characterized by rashes in children's parvovirus. So this is having single-stranded DNA. And again, bacteriophage. Have you heard about bacteriophage?
Yes, I do.
What is it? The
The
why?
bacteriophage, it's like that thing with like the head and like the sticks for like the legs and it like
No,
goes
first
on
of
the
all, what is
cell.
it?
It's a virus. It's a kind of virus.
But why is it called as bacteriophage? It's a virus that infects the bacteria.
Yes, I
That's
guess.
the reason we call it as bacteriophage. And usually T4 bacteriophage, you would have seen a structure like this. Something. Have you?
Yes, that's the scary thing.
Ah, this is the scary thing. Oh, nice. Okay, nice. So this is also having double-stranded DNA molecule. Then herpes virus, which is that you have that infections in the skin with small boil-like structure, that is herpes. All these have double-stranded structure. Please remember for HIV, influenza, corona, rotavirus, and bacteriophage. One more questions you will have. So this is about the, let me move. Do you want to copy something? Shall I erase it or?
Yeah, no, I wrote down everything I needed while you were talking, so it's okay.
Okay. So the viruses that we are talking about, some viruses even have a very little gene, like 4 genes, and some we call it as a giant virus, 2,500 genes. So it depends upon the number of genes are very little to a large. For example, if you take the human, how much is a human genome made up of? How many genes do we have? Any idea?
Oh, 20,000 to 25,000?
Yeah. approximately 20,500, around 20,500. And so when you take viruses, the giant virus is made up of 2,500 virus. And certain viruses are enveloped. Envelope is a cover around the capsid, right? And so the viruses gets the envelope from where? From the host cell membrane. This was one of the questions. From where do they get their membrane from? So they get their membrane from the host cell and membrane when it is released. When they are releasing, no, they give away the membrane also. So this envelope protects the capsid and gives an additional protection. And also
It's a bit
one
like
more,
the it's a bit like vesicles.
does it?
So is it a bit like vesicles?
When which occurs?
Vesicles.
No, when the virus is infecting the host cell, right?
Yeah, but is
At
this
that
whole
time,
thing
it
with like,
acquires the envelope from the host cell membrane.
so it becomes a vesicle?
Yeah.
But like, does it like go through like phagocytosis? Is like that the kind of thing when you have like,
Phagocytosis and all, like when it is released, it takes the body of the host cell. We are saying no. So, for example, this is infecting a host cell. Well, it takes the body of the host cell, right? So basically, it gets its envelope also when it is being released. When it is released into the host cell, it takes up its envelope. That's what is not like engulfing. It's not like engulfing, like phagocytosis as you do. So
oh,
when
is it? Oh, it's because the capsid can't like go through the membrane, go
yeah,
through a
that's the
host cell
reason it
membrane.
forms another envelope. It gets one more.
What do you mean?
Like, for example, how can I say you? Um Now, this is the, let me say, this is the virus which is not enveloped with the protective cover outside. This is the capsid protein, right? And so how does it get its envelope? Like, and this is more stable and resistant to the environmental factors. So when it is infecting a host cell, for example, this cell is having a plasma membrane. So this goes inside. So it gets this envelope. Now, you're getting it. Now, this is the plasma membrane and this is the DNA inside it. When this goes inside, where will this begins to belong to this?
Oh, okay.
You're getting it.
Yeah.
That is what we say it as. Okay. So because of this getting an envelope, what happens is the immune system doesn't recognize it as a foreign cell because it is already knowing this as the one which is present here because of this envelope. It's able to give a disguised feature to the immune system. You're getting it? What I say?
That's why we get super sick and you never know what's happening.
That is the
So that's why like when you get like super sick as like a virus like attacks you or whatever, you don't know what happened because your immune.
yeah, correct. So you do not have an antibiotic in this case only because it is taking up your host cell. It is not, your immune system is not recognizing it as a foreign body entering, right? It is the same cell. It takes up the body of that cell, right? You're getting it. Is it fine? Hello.
Yes. I get it.
Okay. So as I told you, some are enveloped, some are not enveloped. This bacteriophage, lambda and all, and adenovirus and all are not enveloped. And as mentioned, some because of, even though they are non-enveloped, they are stable and resistant to the environment than the enveloped virus. As because this envelope, the lipid envelope, sometimes get damaged due to environmental stress like heat, light, and chemicals, which they are, if they are exposed to it, right? And so when we talk about the HIV, so we know HIV, I think you would have seen the structure of an HIV. Is it an enveloped virus or not enveloped virus? It's an enveloped virus, okay? You would have seen a structure like this.
Oh yeah, because it has like the yeah.
Spikes like something. So inside you have the capsid and outside you have the envelope. So it is AIDS, it causes AIDS in human and it has an envelope and it gets the thing from the plasma membrane, as I told you. So on the envelope, you can see glycoprotein structures.
Yeah.
So using this only, the virus gains entry into the host cell.
Mm-hmm.
So the HIV, unlike the other viruses, it has an unusual, we say it as a cone-shaped, it will be like this, like a tetra one, something like this it will be, basically. So cone-shaped capsid within which it has a single-stranded RNA, and it also has three important enzymes. Do you remember what enzyme?
Yeah, they are biological catalysts that speed up the rate of a reaction.
Yeah. What is the name of the enzyme? Reverse transcriptase.
Oh, that's what, oh,
Reverse transcriptase means
it reverses the transcription from,
what is transcription? Transcription is DNA to mRNA, right? So
yeah.
here RNA is converted into DNA using the host cell machinery. Since you're getting it, usually
Mm-hmm.
this DNA to RNA is transcription. This is reversed. You have a single-stranded RNA which is been converted to a DNA. You getting it?
Yeah, yeah, I
Same
am.
way when we see a SARS COVID 2. It is
Yeah.
also, and it also has a structure like this, but it will be little quite different. I think somewhat like this, it will have a capsid and or a DNA material. So it also has an enveloped one. And so it has single-stranded RNA genome. The virus is enveloped and it has proteins and envelope proteins on the surface. But you can see larger spikes. When you compare AIDS and this one, this has a larger spikes like this, something like this.
And
So
little
and
flowers.
yeah, yeah. So we call it as a crown-like structure. It will be like a crown. So this
I was looking at my notes and I have a question.
pardon.
What's the difference between a spherical virus and a conical virus?
Spherical and conical means I don't see.
The
This
shape.
is spherical. So this is spherical. Like I'm drawing the capsid like this, right? Something like a flower. This is conical. I don't know, some pentagon or something. Okay, hexagon like this is what is considered as conical.
So then what is icosahedric?
Pardon?
Because in my notes I have three like shapes. I have spherical,
Hmm.
conical, or icosahedric.
Hmm.
And like the icosahedric is the phage lambda, the
Like for
scary
example,
one.
can you see my screen somewhere?
No,
Can you
I
see
just
my screen?
see the whiteboard.
Okay, 1 minute. You see the structure. This is what we consider it as spherical. Inside one, right? Can you see?
Mm-hmm.
But conical is, let me go to HIV. Can you see the structure?
Yeah, but I don't understand the difference. Is there just a difference in like the capsid and like what's on the inside,
Yeah, it is capsid only, not the
outside?
envelope.
Oh.
It is capsid only, not the envelope. Envelope is always spherical only. It is inside the capsid. Like envelope proteins mostly are spherical in nature only.
Oh, okay.
Talking about the capsid that is inside, that is capsid, the glycoproteins, the type of enzymes, the genetic material that is present is the one that gets varied from one to another.
Uh-huh.
Okay.
Yeah.
So if you take the bacteriolambda, I'll show you that horror one. This is the structure, right?
Yeah.
This is the structure. So this is not this is not an enveloped one. You can see a double-stranded DNA over here. It has a we call this as I think icosahedral capsid, ICO.
Yeah.
It's the icosahedral shaped capsid head, and inside that you have a double-stranded DNA. It has a short collar, tail, and a tail, and a fiber also attached to the cell. Using that only, they get infected into the host cell, and you have so many enzymes within them. One of the most important enzyme is holin. Have you heard about it?
What to do with
Hole. It makes hole in the cell wall of the bacterium when it is infecting. Holin. Hole.
ah
Okay. So this is about the three things that is mostly in your syllabus. Corona, we take it because so it's a real life application. But HIV and bacteriophage, we commonly study. How is it? How are they different from the others? Okay, now we'll move on to the HL portion, which is lytic and lysogenic cycle, a very easy one. Lytic, what is lysis?
the splitting or breaking of something.
Lysis means mostly, okay, breaking or splitting. So the viruses which we are talking about are made of protein, and they have only one type of nucleic acid, which is either a DNA or an RNA, and they do not contain organelles.
No, they
Right,
don't.
and any cell structure which has the ability to make themselves more in number, that means they cannot replicate the cell structure, they cannot replicate or they cannot reproduce outside the host cell. So we call this viruses as obligate intracellular parasite.
I see.
Obligate intracellular parasite, which means they live on other living organisms. Obligate means restricted to a certain functions. And we know intracellular means they are present within the cell, and we know what is a parasite. They experience the benefit from the host, right? So since they're dependent on a host cell, we give this a name. So why do they depend on the host cell? We know basically for the purpose of energy. So because they do not have mitochondria to produce their own source of energy like other living cells. So for energy, they depend. So that only when they have energy, they can replicate in numbers. Nutrients is the next factor. They don't have any appropriate system. So for building up for air, they require some building blocks for viral replication. Next reason is replication machinery. So they don't have a exact organelle for themselves to replicate. So they rely, for example, ribosomes, enzymes for transcription, translation. Though they have the genetic material, all these machineries are not present. The last reason why they depend is transport. So this transport, they cannot do on their own. They cannot move to other parts of the body since they don't have their own transport mechanism. So these are the reasons they depend upon on the host cell. And so viruses need to gain access into the host cell to take over the host cell machinery, correct? So what are the ways in which they gain access? As sometimes some viruses, like as you told me, they have membrane bound vesicles. So by process of endocytosis, like we call it as an invagination, right? So based like that, they can gain entry into the host cell. The next is receptor mediated diffusion. Receptor mediated fusion. So
So
in the
these are
name,
ways they enter the host cell.
pardon ma?
So these are the reasons why they enter the host cell, how they enter the host cell.
How they?
No, I'm just asking what you're telling me right now, this endocytosis and the receptor.
Endocytosis is like they make a
Yeah,
they
I
approach.
understand that. But like these are ways that
Yeah, they
this
gain
parasite
entry into
enters the
the cell.
host cell,
Yes,
like the
that
viral
is the
DNA.
host. Correct. They access the host cell by these ways.
Oh, okay, okay.
They should have some method of getting into the cell, right? So one is
Mm,
endocytosis. Second one is receptor mediated. The name itself says receptor mediated, which means they bind to the receptor that is present on the host cell. They fuse with it and enter it. That is receptor mediated. So best example which we can take for receptor mediated is bacteriophage. So if you take receptor mediated, we know that the membrane contains receptors, right? So these bacteriophage will bind into the receptors which is present on the surface. As a result, they just pass their nucleus into the host cell. This is the receptor mediated. If you take endocytosis, it is like this. So they form a vesicle inside and they just release their nuclear material outside. So these are the two methods by which they gain entry into the or get access into the host cell. Endocytosis and receptor mediated fusion. Is it fine? Is it okay?
Yeah, it's okay.
Okay, talk about lytic and lysogenic cycle, what you have learned so far.
Well, I learned it, I just don't remember it. All I know is that one is active and one is dormant. And then at one point, the lysogenic cycle.
What is this lytic and lysogenic cycle? What is it about?
I think there's something to do with replication, right? Replication virus.
Reproduction. Genome reproduction inside the host cell. So now we
that
know how they gain entry inside the host cell. But as you told, the replication should happen. It should undergo replication. It should produce in numbers to get it themselves spread. So some viruses use this mode of reproductive cycle, which is lytic, and some use lysogenic. And some switch between lytic and lysogenic. That is also possible when they are exposed, especially to some special environmental condition like UV light or chemical stressors. As in that case, is you have both lytic and lysogenic happening. Example is bacteriophage. Lambda. Bacteriophage lambda. So this is the one that infects the bacterium E. coli. So what happens in this lytic cycle? So the bacteriophage will take up, we know it takes up the host cell machinery, right? And so the virus starts reproducing. So how does this virus start reproducing? The viral particle burst. You told me split, right? So lyse, which means they lyse or burst the host cell, completely destroying the host cell.
Okay.
For example, if I think the diagram will be like this. So this is the host cell DNA, which I'm drawing. So when this bacteriophage infects over here, you have the bacteriophage DNA entering inside like this. So now it has entered the lytic cycle. Once it has entered the lytic cycle, you can see that this bacteriophage gets assembled inside the host cell, many in number. So when they are released, what happens is completely destroys the cell. So the cell becomes destroys and all these bacteriophages are released. Bacteriophage will lyse the cell, causing the new viruses to be released from the cell and spread outside. This is lytic cycle of bacteriophage lambda. So this is what happens in lytic cycle. So the host cell is completely destroyed and you have the new virus particle being produced. On contrast to the lysogenic cycle, if you take here, the viral particles are not produced as well as the host cell is not destroyed. So it takes place for a longer period of time where the viruses remain in a dormant state.
Okay.
So when in lysogenic cycle, what happens is when the bacteriophage enters the host cell, it becomes integrated with the host genome. And as the host cell produces, so once it is integrated, right? So once as this cell is producing, it will produce a cell with the bacteriophage, which is called as prophage. So it will have its own genetic material as well as the bacteriophage genetic material. You're getting it. Are you able to understand what I'm trying to say?
Yeah. One of them,
like this. Yeah, it will have
completely
like
destroys.
Yeah.
The lysogenic does not destroy, and the bacteria and the host, like the bacteria DNA, stays in a dormant state.
Yes. So this is the fused one. So this is the bacteriophage DNA. This is the host cell DNA. So when this divides, what happens? It will carry both. So bacteriophage DNA also will be carried. The infected DNA also will be carried. This also. So same way it gets reproduced. So sometimes, and so we call that, that's why this one, no, which is being produced, we call it as a prophage or prophage. So the bacteriophage DNA then integrates within the cell. So once they get divided, both will be produced. The viral DNA will, so there is no destruction of the cell, neither the new virus particle being produced. That's why we say this is a lysogenic cycle, and we say that viruses remain dormant.
So then how does the lysogenic cycle affect our immune system, for example?
Pardon, can you come again?
How does the lysogenic cycle affect, how
See
does
here,
like,
can you see this diagram?
yes.
Can you see the integration happening and the production? Here also there is production, no?
Yes, but how does this affect our immune system? Because it's in the same host cell. So if an immune
It is in the
cell,
host cell. That is fine, but it is carrying a virus, right?
yeah,
Carrying a viral DNA. Am I right?
yeah,
So isn't it infectious again?
I guess
Here
so. But
also the
then what,
virus is being,
but then I
tell
feel
me,
like,
tell me.
for
This is
example,
more
if our
better
immune
than this.
system attacks these cells, right, then it's
See, now
also
this is
killing
only
the,
again entering the lytic cycle. This is how they switch. That's why we
like what?
say it is in a dormant state. It is making up. There is no, what to say, like lytic cycle, you don't have any uh, increase in population or larger effects. That is not there. The lysogenic cycle just involves the fusion of the viral genome into the host genome. That is what happens. So it is just lying dormant within the cell, involving in only the fusion of the nucleic acid of the bacteriophage with the host cell. This will be followed by the proliferation.
Oh, okay, okay, okay.
What is happening?
I think I get it now.
Okay. Shall we move forward?
Mm-hmm.
Okay, then the next is the origin of evolution of virus. So in the beginning itself, we told viruses are of great importance because they are a very important evidence of evolution, and we saw that viruses exist in various diversified structure and genetic material. So it is assumed or it is suggested that they come from various possible origin. So where did that originate from? So according to many studies, it is stated that viruses could have come from the genetic material, the ancient DNA or RNA, just like that, encapsulated over a protein coat. So we call that as protovirus. So it may be just the RNA coated with a protein, which would have evolved into viruses, what we call now. And as I told you in the beginning, I spoke about viroids. So there is also researches or there are also scientific findings saying that the viruses could have evolved from this basic unit of particle, which is small infectious agents, which can be lesser short strands of RNA alone, on which was found to be infecting some flowering plants. So it would have also been evolved from this viroids. The next findings is transposons. Have you heard about transposons?
No.
Okay. Transposons are, again, some small segments of DNA or RNA, but they have the ability to move position within the genome. They can move position within the genome. That is
Mm-hmm.
what we call it as transposon. So there are studies saying that viruses could have evolved from transposons by moving their positions within the organism's genome. In that way, also they could have been evolved. Sometimes some findings state that viruses may have evolved from ancient cells, like which didn't have the ability or which has lost the ability to live independently. Like they have no other option, like depending on something and living for a continuous period of time. And so it would have also been evolved from such organism is also the study state. So it may be from an RNA or DNA just encapsulated on a protein, from viroids, which are small infectious organism, from transposons, and from some organism which didn't have the ability to live independently, right?
Uh-huh.
And so
Uh-huh.
usually we consider this evolution as convergent evolution. What is convergent evolution?
Okay, so convergent evolution is like there is no common ancestor, right?
No, no common. Yes. So
No common
distantly
ancestor.
related or yes. So they would have acquired some similar trials in terms of adapting to the environmental conditions, right? Though that is called as so unrelated organisms. So they say that unrelated organisms could have got similar trials by adapting to some selective pressures. That's the reason this viruses also can come under the category. Why? So some viruses which we are studying infect different cells, as and we saw that the access methods are also different. And so that may be a reason. So they would have all combined some similar patterns. Some viruses, we saw that they evade the host cell and they're trying to spread the infection. Some viruses infect different types of host organisms. So based on all these characters, they could have grouped themselves, is what is the study says, right?
Mm-hmm.
Okay. So, but we know, though the studies are saying that and this, there is a rapid rate of evolution in virus, is what most of the researchers have given us the fact. Viruses evolved at a faster rate because of their taking up the host machinery. So many RNA viruses, especially these retroviruses and all in the HIV, has a higher mutation rate.
I
So,
see.
they have because you see they can copy the genetic material, this reverse transcription enzyme and polymerase and all. They can easily mutate themselves at a faster rate
Mm-hmm.
when you compare with that of other enzymes like DNA polymerases. That's the reason HIV, influenza, and all, you can see them spreading very fast. So the infections, and it's up to 10,000 times when you compare that with the DNA viruses. RNA-based viruses have a higher mutation rate. And this mutation doesn't stop with themselves. They pass on to the next generation of the viruses also, right? And so in that case, we say that there is rapid evolution happening. And as we saw that viruses exchange genetic material, we saw recombination happening, like the virus genetic material and the original genetic material combining together, gene transfer. I get that characteristic from the host cell. In that way, they get evolved. And this viruses have very short generation, but the reproductive rate is very high. That is also a possible reason for evolution. As I told you, the best example is the evolution in HIV. Some, somewhere around that time only, 1970s or 75 to 80. This got transmitted mainly through the body fluids, which is the semen and the other stuffs, right? And so when we talk about this retrovirus, it infects CD4 plus cells. Have you heard about this? CD4 plus cells?
Nope.
Okay, it's T helper cell.
Oh, that I've
In,
heard of it. I've heard of T helper
yeah.
cells.
So they are a part of a very essential part of an immune system. So this retrovirus was directly infecting this CD4 plus cells, leading to this acquired immunodeficiency syndrome. It is a pandemic one which we saw, which has been spread very worldwide, right? But now there is a cure recently. But when you see, until the previous days, we didn't have a proper treatment mechanism, yeah, right? And because there was a high rate of even corona, what happened? We faced so many evolutions. Till now, we are having so many strains of COVID, right?
Yeah.
Because of the high reproductive rate. So each time they get evolved, the mutation rate is very high. They change their genetic material. And that reverse transcriptase enzyme, it is more error prone. when you compare with that of any DNA polymerases. So this has led to virulent strains, like more harmful and toxic strains of this. That is the reason they were unable to find an antiviral drug. And as we saw, they combined with the genetic material of the host cell. So you have to
Yeah.
destroy the genetic material of the host cell to destroy that virus, which is very difficult. Right?
Yeah.
And same way, influenza virus. You would have heard about flu. Commonly, that influenza is flu, which is also an RNA virus, as it has a less accurate means of replicating when you compare with that of the DNA virus, but high mutation rate. It has two important stuffs, which is heme agglutinin and neuraminidase. Neuraminidase surface. So it is made up of that protein, heme agglutinin and neuraminidase surface protein. So that is, these two things make the immune system recognize them very it becomes very difficult for them. It's like a protection to the virus. An immune cell cannot identify this as some foreign substance. And that's the reason it took so much of time to have vaccines for influenza also. Oh,
Oh, okay.
yes.
Yeah.
And one more important characteristics of this influenza virus is they carry out a process called as reassortment. Reassortment. So when this virus is infecting a host cell, right, and like which already has some strain of influenza, which means it is already infected with one strain. So when a new strain is infected, what happens is the viruses, the host cells has two different genetic material.
Yeah.
You're getting it? So this leads to a new strain. This is reassortment. You're getting what I'm trying to say?
Yeah, I get it.
So basically, this has a high evolution rate. Why? Because it keeps on changing from time to time due to the high mutation rate within the enzymes. That's what we are trying to say. I already spoke about the coronavirus, which happened now, the SARS-CoV-2. It had a high replication rate. It started copying itself differently at different period of time. It started mutating itself and getting accumulated. And we had SARS-CoV-2 Delta, then we had Omicron variants, and so many things came one after the other, which made us difficult to develop a vaccine. But anyhow, we came out with vaccines and we had something happening.